So how, are you ready?
Yeah, I can be ready, sure.
Okay, let's go. Hello, everyone. I'm very happy to see all of you here. It's a real honor to be the host for this session. We have had a stimulating session this morning. Now we have 5 distinguished speakers: Professor Hao Wu, Professor Wenhong Zhang, Professor Qiming Zheng, Professor Yi Pan, and Professor Zibo Jiang to give the keynote speeches in this evening session. They are here to share some amazing insights from the research. It's my great honor and privilege to introduce our first keynote speaker, Professor Hao Wu. Professor Hao Wu is affiliated with the Children's Hospital Boston, which holds a special place in my career as my former workplace. Professor Hao Wu is a prominent biochemist and a Christian logographer. Professor Wu is an endowed professor at Harvard Medical School. Professor Wu's research is well recognized, and she was elected a member of National Academy of Sciences in 2015, and elected a member of American Academy of Arts and Sciences in 2021. Professor Wu conducted her postdoctoral training at Columbia University and obtained her PhD at Purdue University. She was trained at Peking University and later Peking Union Medical College in China before she came to the USA. Professor Wu has 216 publications, many of which are published in leading journals such as Cell, Nature, Science, etc. Specifically, she has 23 papers in Cell, 23 papers in Nature, and 15 papers in Science. Today, she's going to guide us through a very important topic, inflammatory stomach. Hao, it's yours. Thank you very much.
Ursi, thank you very much for the introduction. And thank you for inviting me. I am delighted to present our work among so many distinguished speakers. So my lab has been interested in using, I would say, primarily structural biology to elucidate fundamental questions in immunity, in particular innate immunity, which offers the first line of defense against infections and other types of danger. Our initial work began from the TNF receptor superfamily, which then migrated into toll-like receptors. And more recently, we've been focusing our work on inflammasomes. Trying to get a laser pointer. So it is through our work in these different receptor systems that several years ago that we began to realize that in these systems, the aggregated molecular assemblies are actually the functional forms. And this was anti the concepts that was the norm at that time. For example, we've observed these helical assemblies that's now everywhere for these proteins in the so-called death fold domain superfamily. They form these helical assemblies so that they can propagate their signaling in a way that's different from traditional signaling by receptors such as G protein coupled receptors. We also observed these multivalent assembly. For example, if you have both dimers and trimers in your domains, then they can come together and they can form these infinite molecular assemblies, if you will, especially on the cell surface. We've also observed.
Could you please share your screen?
Oh, you did not
Yeah,
see
sorry
my
about that.
screen. Oh my God, I didn't realize that. Okay. All right. Share screen. Oh, where is it? Okay. How's that? Now it's better?
Yes,
Can you
great.
see my screen now? Okay,
Yeah.
sorry, but that should have told me earlier. Okay. That was just my first slide about the different receptor systems that my lab has been working on. So these are the type of structures that we have observed over the years, from helical assemblies to these multivalent assemblies to even amyloids that's driving the aggregation of the protein and therefore their functional aggregation, activation in cells. So this kind of signal transduction, we proposed that is different from a classical view of signal transduction in which ligand binding induces conformational change in the proteins inside the cell and therefore generate second messengers and therefore activate the cellular response. In the types of innate signaling pathway that we have observed, the receptors, the ligands, substrates, multiple substrates of these proteins and their adapter proteins all coalesce into these molecular assemblies, and therefore they share certain interesting mechanistic similarities to other ways to assemble high order structures such as phase separation. So it is in this context that we started to work on inflammasomes, which are supramolecular complexes that activate caspase-1 or other inflammatory caspases such as caspase-4 and 5 in humans and caspase-11 in mice. So my lab has done quite a bit of work on how these inflammasome sensors senses their ligands and then form the molecular platform for caspase-1 activation. So upon caspase-1 activation, the number of substrates can then be cleaved, and this include, I would say, most prominently cytokines in the interleukin-1 receptor, interleukin-1 superfamily, such as interleukin-1 beta and interleukin-18. So even though this is a very old subject, how these cytokines gets activated or processed by caspase-1 or other inflammatory caspases remained unknown. So another breakthrough in the field was the discovery of this pore-forming protein known as gasdermin D. Upon cleavage or other types of stimulation, the N-terminal part of the gasdermin D protein is able to form pores, and this was we and others have discovered this back in 2016. And these pore structures is important for the release of these processed cytokines to the outside of the cell to induce inflammation. It can also induce or start to induce this lytic cell death known as pyroptosis. But more recent work suggests that gasdermin D itself is not sufficient to induce the cell death, although it is sufficient to release the cytokines, but not sufficient to induce the membrane disruption during lytic cell death. NINJ1 is a protein that appears to be acting downstream of gasdermin D and execute this final step in inflammasome activation. So in today's talk, I will focus on these two aspects of inflammasome biology. They're currently unpublished. And as I will show you, for both of the subjects, the insight that we obtained were quite unexpected. So let me start from the maturation pathway for pro-IL interleukin-18. which may also be true for interleukin-1 beta, but our structure data are on pro-interleukin-18. And this is a true team effort. My postdoc in Dong led the project, and the group of Louis Kay at University of Toronto determined the NMR structure of pro-IL-18 by itself, and Pascal Devon in John Kagan's lab in our Boston Children's Hospital helped to perform certain biochemical and cellular biology experiment. And Johnny Liang helped Ying to purify some of the proteins when she was pregnant with their first baby. Johnny is the husband of Ying Dong. So all of them collectively contribute to this project. So if you look at this, this is an in vitro cleavage of caspase-1 by caspase-1 of pro-interleukin-18, pro-interleukin-1 beta, and as well as pro-IL-37. This is also a member of the interleukin-1 cytokine family, and also gasdermin D. So gasdermin D is the, I would say, the best substrate among all the substrates for the caspase-1 family. And if you look at whether caspase-1, this is the catalytic mutant, so that you can form a complex with the substrate without cleaving them. So if you see these two proteins do form a very nice complex, and these were individual traces of caspase-1 and pro-interleukin-18. And when you put them together, they have a very large shift in the elution position, suggesting that they are forming a complex. And as I will show you later, this complex formation also drives the dimerization of caspase-1. So you may know that caspases are activated by dimerization. Monomeric caspases are not active, only dimeric caspases are active. This was work that my lab has contributed previously, but Yigong Shi's lab has also set the concept for this caspase-1 dimerization. So cryo-EM structure then of the complex between caspase-1 and pro-IL-18 was determined. As you can see from these two-dimensional, these are just projection images, two-dimensional images of the complex. They show up as these candy-shaped little structures that are kind of cute. And if you perform 3D classification to look at how they look like in 3 dimensions, you see these different kinds of assemblies, but the major class is this one that counts for about half of all the particles that we obtained in these cryo-EM micrographs. So here I show you the cryo-EM structure of caspase-1 in complex with pro-interleukin-18. This here is colored by resolution. So the overall resolution is 3.2 angstrom, and the resolution range goes between 3 angstrom to much more disordered or lower resolution. So the caspase is very ordered, and so this is a caspase part. This is a dimer of a caspase, and this part that's contacting the caspase of pro-interleukin is also very well defined. So in here then, we show you the structure of the complex in domain colors or molecule colors. This is the P20 of caspase-1. P20 is the large subunit of caspase-1. And then these cyan color and this mauve, if you will, mauve color is the small subunit, the 2 of the small subunit. And this again is the large subunit. So the interaction between pro-interleukin-18 and caspase-1 has 2 interfaces. So one is the interface in which the cleavage site, right? The cleavage site directly goes to the active site of caspase-1, which is around this position here. And this is how caspases usually recognize their substrate. But in this complex, there's another interface, and this interface is away from the active site, not at the active site, but away from it. And this, as I will show you, is an important interface that results in the recruitment and capture of the pro-cytokine in order to process it. So similarly, we also obtained the structure of the caspase-4, which is another inflammatory caspase. And this molecule is also the lipopolysaccharide receptor. So LPS directly binds to caspase-4 and activate caspase-4. So we see a very similar structure of how pro-interleukin-18 gets processed by caspase-1 as well as by caspase-4. Feng Shao's group also obtained this particular complex. So if we look at the interfaces and try to design mutations that will disrupt the interaction at the active site or the exosite, you see that they compromise the release or secretion or the maturation of the interleukin 18 from cells. These are THP1 cells that are reconstituted with these caspase-1 mutant, either at the active site or the exosite. The background is a caspase-1 knockout. So you see they reduce the amount of activation that's in cells, and in vitro, these caspase-1 mutant also compromise the cleavage. So these are KCAT over KM that's determined from in vitro cleavage. You see some of the mutations are greatly impaired in their ability to process pro-interleukin-18. Similarly, if you mutate these interfaces from the site of interleukin-18 by the wild-type caspase-1, you also see certain defectiveness in this processing. So I want to draw your attention to this panel here. This is a caspase-1 processing of pro-interleukin-1 beta. So if we use the same mutations in caspase-1 that's defective for interleukin-18 processing, they're actually also defective in pro-interleukin-1 beta processing, suggesting that the mechanism of processing of interleukin-18 versus interleukin-1 beta may be the same or similar. So here I should also show you an example in which if you mutate the caspase-1 interface with pro-interleukin-18, they no longer form a complex. As you can see here, they no longer have this shift in the gel filtration profile. So that was nice, showing that there's this two separate interfaces that's important for the capture of pro-interleukin-18. And if you're you may have remember that Feng Shao's group and Sam Xiao's group previously have also shown that caspases can capture gasdermin D using a similar mechanism, because they also have two sites, an active site and an exosite. But the dependence on the degree of dependence on these two sites were quite different between gasdermin D and the cytokines. So we were additionally curious about why pro-interleukin-18, which has a pro-domain, right? Pro-domain followed by the mature domain and with a cleavage site in the middle. What is the mechanism why this protein is not active against, is not active for activating the interleukin-18 receptor? So in order to address that question, we determined the NMR structure of pro-IL-18, not in complex with caspase-1, but alone by itself. So here I show you the green represent the pro-domain and mature domain is shown in orange. So these are two views, and the tetrapeptide or the sequence where it gets cleaved is somewhere over here. So surprisingly, this pro-interleukin 18 structure is not a bead on the string. It's not like it has two domains and then with a flexible linker in the middle. By contrast, the two domains are really integrated together into a compact structure. And this is a structure that gets recognized by Caspase-1. There are certain minor differences between the conformation of pro-IL-18 alone versus its conformation in complex with Caspase-1. For example, the tetrapeptide motif that gets cleaved is somewhat different between these two structures, and certain local conformational changes also occurred. But overall, they're quite similar. However, though, the pro-interleukin-1 has a structure that is completely incapable of receptor binding. And the reason is that the region that the mature interleukin 18 uses to bind to its receptor, this magenta region here in the mature interleukin 18 structure, is not formed at all in the pro-interleukin 18 structure. So there will be a massive conformational change between the pro cytokine and the mature cytokine in order to build this receptor binding site, and therefore explain why only after cleavage mature IL-18 can bind to its receptor. So as a summary, then pro-IL-18 dimerizes caspase-1 to activate it. So what do I mean by this? So if you remember how caspase works, so caspase-1 has an N-terminal CAR domain. So these are caspase recruitment and activation domain. The CAR domain forms a filament, and on top of the filament, the caspase domains would have a very high local concentration. And the concept of the field has been that these caspase domains would be able to dimerize in the context of the high local conformation. However, in our this recent study, it shows that these caspases are actually remain to be monomer, and it only become dimerized after substrate binding. So this explains why the caspase molecule is only active at the inflammasome spec, the inflammasome center, where everything comes together. And this because these monomeric caspase, once leave the scaffolding protein, can no longer become dimerized by their substrates because of the low local concentration. And pro-IL-18 is a compact structure, which is poised for recognition by caspase-1. And then IL-18 refolds after cleavage to build this receptor binding site in order to become active. And therefore, the small molecule, fairly small protein of pro-IL-18 really is regulated by several steps, and the entire conformational transition need to happen before the generation of this highly inflammatory cytokine. So in the second part of my talk, I would like to present to you our recent work on NINJ1. So this is a protein, a transmembrane protein, if you will. It has two predicted transmembrane regions, TM1 and TM2, and it has an extracellular region that tend to form helical structures. So I will show you in our structure, this is actually two different helices that are band next to each other. So this protein was initially identified as a adhesion protein that's activated during for example, when neurons are damaged, and this protein is important for neuronal repair. So in order to look at how NINJ1 can damage cell membrane, we reconstituted the NINJ1 and also NINJ2, which is a related protein that is highly similar but cannot damage the membrane. So this is like a negative control. So when we reconstituted the protein and purify it through a sucrose gradient, you can see it start to form these ring-like or sometimes filament-like structures, but are highly irregular. They're not completely ordered, highly irregular. NINJ2, however, does exactly the same thing. So this doesn't really distinguish NINJ1 from NINJ2. So in order to look at how NINJ1 could exert certain functions on the membrane, we first looked at how NINJ1 could bind to different kinds of lipid. This is a lipid strip assay. As you can see here, it has affinity to PI4P, but NINJ1 and NINJ2 both have affinity to PI4P. And this intermediate in lipid biosynthesis, phosphatidic acid, can also bind to NINJ1. So the striking thing is that when we mixed NINJ1 with liposomes, so this is how a liposome will look like. When we mixed NINJ1 with liposomes, the liposomes are simply gone. They are dissolved into tiny little pieces, which in a way recapitulate the function of NINJ1, that is to damage the membrane. NINJ2, however, when you mix NINJ2 with liposomes, if you look at the edge of the liposome membrane here, they also bind to the membrane and even form oligomeric structures, but they do not cause the rupture of the membrane. Here I show you purified NINJ1 oligomers from the liposome preparation. So here is another image showing that these are liposomes before mixing with NINJ1. And after you mix with NINJ1, they become much smaller, and essentially NINJ1 dissolved the liposome. So we were able to use a fairly unique method, if you will, to solve the structure. So what we did was to take these large NINJ1 rings and just cut out little pieces so that they become a bit more homogeneous. So these are 2D images of these little segments. So the length of these segments is about 6 to 8 subunits, and they are part of these linings in the ring structures that we observe for NINJ1. So then we were able to obtain a cryo-EM structure of the NINJ1 segments. So interestingly, these NINJ1 segments all have a curvature. And as I will show you later, the curvature always faces the same way. So which way does it face? So this way, the way it faces is actually the hydrophobic surface of the NINJ1 molecule. Whereas on the other side here, the surface of NINJ1 is highly hydrophilic. So which means only this side is compatible with membrane, whereas this side is not compatible with the membrane. So given the way that the assembly can be formed, which is this I will show you in the next slide, that is to put the strand, the extracellular strand, actually insert into the membrane at the region of the next subunit. So you have this chain formation between the subunits through the insertion of the extracellular helix. So if that's the case, there could also be smaller rings that you can build through this mechanism because the entire assembly is fairly flexible. So here I show you the structure of the subunit. The 2TM transmembrane subunits are highly curved, shown like that. And interestingly, glycine residues, which tend to localize kinked regions of helices, localized exactly at these kinked regions of the TM helices. And this is the extracellular domains. They're supposed to be extracellular, but once activated, they insert into the membrane. And this is what we think the mechanism by which it cuts through the membrane. Because it's hydrophilic, it has to repel the membrane and therefore can cut through the membrane. So previously, another group has recently published their structure of NINJ1. So in their NINJ1 structure, they have double filaments. So what they have, instead of having ring-like structures, they have two filaments that stack together. And these are very straight filaments. And perhaps because of the stacking, which we think is an artifact during the protein preparation, the helix, these are the two transmembrane helices. The helices are straight rather than kinked, and there's no way to tell which side of the assembly faces the membrane versus faces the hydrophilic part. I show you the double filament 1 and 2 here. So in our case, if you look at all the 2D averages as shown here, you always see a high density region and the low density region. And always the high density region is the concave side, which is facing the lipid, whereas the low density region is the convex side, which faces the outside of the membrane, suggesting that these rings always have lipids inside. At least that's what the structure would tell you. And I will show you some evidence that this also happens in cells. So we used structure-based mutagenesis to look at certain residues in NINJ1 that's important for its function. These glycine residues at the kinks, when you mutated those, they are no longer active, shown by these magenta colors here. They have much reduced ability to induce cell death. We also look at these amino acid that faces the hydrophilic side, as well as amino acid faces the hydrophobic side. And finally, we looked at a cluster of positively charged residues that we think could be binding to PI4P. So all of these mutations are defective in cell death. And I want to show you these potential PI4P binding residues. So interestingly, these residues changes the localization. Changes the localization of NINJ1 rather than primarily residing on the cytoplasmic membrane. These mutants are mainly localized or accumulated inside the cell. This could also then support that this cluster of residues is important for binding to a certain type of lipids that determines its destination in the cell. So without being on the cytoplasmic membrane, it will not be able to disrupt the cytoplasmic membrane and therefore have reduced ability to induce membrane rupture in cell death. So we then reconstituted NINJ1 knockout THP1 cells. These are monocytic cell lines, and differentiated them into macrophages. And in this time-lapse movie, which I will also show you the movie, you will see that there's these small structures, sometimes ring-like, that are formed on the cell surface over time, and then they get released to the outside of the cell. And this is understandable if you have a protein that acts as if it's a cookie cutter. You cut out the membrane, and of course they will get released to the supernatant rather than stay with the cell because they're no longer attached to the cell surface membrane. So if I can play this movie here, right? So you see, as you progress in the apoptotic pathway, you see the cell essentially disintegrate and they release these ring-like structures. Oh. So do we have other evidence to support that these NINJ1 oligomers are actually released or shed to the supernatant? So this is a non-reducing SDS page after the activation of the inflammasome. So as you can see here, when you, so these are DOX inducible lines, so you need to induce the expression of NINJ1 and prime the cells for inflammasome activation and then actually activate the inflammasome. So after activation, NINJ1 is no longer, or very little NINJ1 is still associated with the cell pellet, with the cells. Instead, they're released to the supernatant, and they're released to the supernatant in the form of oligomers, because these are non-reducing SDS page. Somehow it preserves the oligomeric structure of NINJ1. And if we take the supernatant, also image them using electron microscopy, it's pretty remarkable. You see these disc-like structures that could be little membrane patches that's floating inside this in the supernatant. But how do we know whether these disc-like structures are NINJ1 associated structures? In this EM method, of course, we wouldn't know, but we performed this alternative experiment, which we think is really a killer experiment, if you will. So NINJ1 here is labeled with GFP. This is also the supernatant of THP1 after inflammasome activation. So you see that these NINJ1 localized to the periphery of these disc-like structures. But when you also stain for lipids using this FODP dye, it's a lipid staining dye, you see that lipids are also inside these ring-like structures, as you can see for all the different structures in the supernatant. So this further support our hypothesis that NINJ1 form a belt around a patch of lipid, and then it just cuts it out and release it to the outside of the cell, which provides an elegant mechanism to damage the membrane. So here's a summary of how we think NINJ1 gets activated. So before activation, the extracellular helical region will be somewhere not inside the membrane, maybe peripherally attached or binds to the membrane, or maybe just in the extracellular space and performing its function in cell adhesion. And upon activation, so we ordered the field, I think, we currently do not know how this activation of NINJ1 occurs, but nonetheless, upon its activation, the extracellular helix, which is amphipathic, has one side hydrophobic and the other side hydrophilic, will cut into the membrane, form an oligomer, as well as then at the same time repel the membrane at the outside of the ring, and therefore form a ring-like structure that cuts out the membrane. Membrane will be inside, and these rings will be released. So we do not have a high resolution structure of the entire ring at this point, but it will be interesting to determine how the lipids really interact with the ring. So my student called NINJ1 a ninja. So it's like a little ninja that cuts out lipids and really kill the cell by completely disassemble it. So this mechanism is very different from what has been published. Because their structure is a straight filament, they had no way to know which side of the filament, when they form a ring, would face the inside. So they had proposed that these NINJ1 oligomers act as if they are also gasdermin D pores. Gasdermin D form this hydrophilic conduit for cargoes to get released. They propose a similar mechanism for gasdermin D, but our data would suggest that NINJ1 use a completely different mechanism for damaging the membrane. So there's so we propose a two-step hypothesis in pyroptosis induction. First, the gasdermin D pores are formed, and at this step, cytokines, processed cytokines, and maybe other things, including metabolites, can be released to the outside of the cell, even if the cell do not die. But on the other hand, if the cell is destined to die, gasdermin D pores delivers a signal to activate NINJ1. We do not know what the signal is, but the signal and NINJ1 activation should lead to this death of the cell by 1,000 cuts, by cutting out little pieces, and therefore can no longer function as a cell. So with that, I'd like to thank all the wonderful postdoc and students who performed these two pieces of work. I did not mention for the first project, Steve Gigi's lab performed cross-linking mass spec for us, and all the cross-linking mass spec data agrees perfectly with the cryo-EM structure. And for the second project, Benjamin Steinberg's lab imaged using super resolution the NINJ1 oligomers on the cell surface, which also agrees with our hypothesis. And I thank you for your attention.
Great. Thank you very much, Professor Wu, for your enlightening and comprehensive talk. We have
Thank
a
you.
few minutes for Q&A. Are there any questions from the audience? Oh, I have a question for you. So you talk about a lot of the very insightful details. I have a question. How do inflammasomes distinguish between harmful pathogens and the non-harmful stimuli?
Right, it's a really good question. It's a really good question. So maybe I can use an example of NLRP3. So NLRP3, or maybe NLRP1. Let's start from NLRP3. So all the things that can activate NLRP3 somehow cause certain damage on the cell membrane. So nitrorising is the one we used throughout our study here. It's a toxin that causes potassium efflux. I think it senses some effect or perturbation to the cell. So rather than sense certain pathogen or pathogen-associated molecular pattern, oftentimes the inflammasomes sense the effect from that, not the direct molecules. If they do not cause potassium efflux, for example, then you will not activate NLRP3. But if you have these pathogenic stimuli that cause a perturbation in the cell, then NLRP3 would respond. And similarly, you can have endogenous or damage-associated molecular patterns, DAMPs. For example, uric acid crystals. So these extracellular aggregates, including crystals or amyloids, they can all be endocytosed, because these are macrophages, endocytosed, and damage the lysosomal membrane. And once that happens, inflammasome also get activated because they sense the lysosomal damage. So in that way, NLRP3 is associated with so many different human diseases, including gout, perhaps through the recognition of uric acid crystals, cholesterol, they recognize cholesterol crystal induced lysosomal damage, leading to its association with cardiovascular diseases or amyloids. So NLRP3 has a really important function in exacerbating dementia, Alzheimer's disease, and Parkinson's disease. Does that make sense?
Yeah, thank you so much. Yeah. Is there any other question?
Okay.
If not, thank you very much again. Yeah.
Thank you. Yeah, thank you for having me. Yeah, enjoy.
Yeah. Okay.